So if you listen to our earlier video where I explained to you how to access IUCr derived annotations for the PDBe web pages, you're also aware that we supply PDBe API endpoints through which you can access these annotations. So if we go to our example entry page again, 5cxt, you can find a link to our API documentation here. By clicking on the link, you end up on a new web page, and here you can see all the different API endpoints grouped by sections that you can access for PDBe information. So here in the section PDB, as well in the section UniProt, you will be able to access the IUCr supplied annotations programmatically. So if we click on PDB, we end up with a whole list of PDB API endpoints which we can use. And here are the two that are relevant for our text mined annotations. If we click on the top one here, this is from a more global endpoint. So here you can find a short description about this endpoint, but also you can see here it takes a PDB entry ID for the querying. So down here you can get a short overview of what this endpoint returns. And just for the fun, you can use the try it out button. You have a preset example and you only need to press execute and you will receive a result. And you can see here now for this structure, we've received the result of annotations. You can either copy them to your clipboard here, or you can download them directly as a JSON file. So if we supply as before our 5cxt example and we execute that query, it takes a little longer because it has more annotation than the initial example. And then we receive here the list of annotations, and you can see, so IUCr supplied the annotations. Here is the DOI to our article again. It is indeed a primary citation, and off we go. So important to note here is that these annotations have been gathered across a number of publications, so they don't strictly have to be for the primary one. So what we have here is everything that is listed that we have for the PDB structure 5cxt. If we try our second endpoint down here, this is the one where you can specify a chain and a residue. So we have a look at this one and we try it out. So here we give 5cxt again, we leave it with chain A. And important to know here is that we need to provide not the residue numbering that we've seen in the article, but the one that is found within the PDB file, within the structure file or the mmsif file, whichever one you use. And in our example, I believe it would be 79. And so if we run that, we hopefully get a result and not an empty dictionary. Yes, here we go. And we have it for the UniProt residue 495. So that's a tryptophan, and we've seen that before in our electron density display. So as I said, there is also the option to look at aggregations in UniProt. So we group by UniProt entry. So here you can see our two endpoints, and if we click on it, we find that we have to supply a UniProt accession code. So if you go for try it out, and for our example, the accession here we use is Q8 VH51, and if we run that example, we get indeed our structure 5cxt, and we'll find the annotations, all of the annotations actually for this UniProt entry. And if you remember, we had 5CXT, but we also had a second structure that was available for us. And if we go further down, that's a long way down. Eventually, hopefully, we'll find the second entry we had, which was, second entry did we have? It was 3S6E, so there should be a second one. PDB, index of PDB, where's it gone? So please, by yourself, have a go and try this out and find your second entry. It's a lot to scroll through. It's a long list. Don't know if I can find it for this tutorial, but you may explore that yourself and see what you can find. So, and if we go to our second option, what we have here is we can also specify a residue, and in this case it's not the one from the PDB structure, from the PDB file or the zip file. Here we have to give the one that is the UniProt residue. So if we go back again, so we use our Q8BH51, and then we specify here, shall we try the tryptophan again, which was 495. And if we execute that query, we should get the details. So we see in the PDB index there's residue 79, for UniProt is residue 495. It is part of the structure 5CXT, and then we get all the annotations for this residue again in each and every chain. So now this is using an interface, but we can also do this more programmatically. So here we have a small notebook which we supply with this publication, and here you can have a read through with a quick explanation of what this notebook does. It allows you to extract the very same annotations that we've just seen through the API interface programmatically. So let's just run all of those different cells with code. So let's first import all the necessary dependencies. Here we have a function that corresponds to our first API experiment. So it just takes a PDB structure, a PDB entry code as input. For this one here is our second example, where we additionally to the PDB entry, we also specify a chain and a PDB residue. We have another one that lets us try out how what we get with the UniProt accession, and our last example uses the UniProt accession and the UniProt residue. And if we go down here, so here are the examples. So I've put some dummy input in already so that you can just give it a try. So if we just run this one and retrieve the results for 5CXT, if you would have downloaded from the interface, from the API interface, the results, you could now compare the two files. Again, you have a JSON result, and you can if you implement this in a piece of code, you can directly save this as a JSON to your disk, to your computer. And here's another one where we specify the chain and the PDB residue. And again, you get an output as before. And you could have compared that if you've downloaded and saved it with what we got from the user interface. And here for a UniProt accession, there we go. Again, produces us a result with the annotations. And the last one, a uniprot accession with a specified uniprot residue, and it is, as ever, our tryptophan 495.